YouTube 这边应该设置好了。嗯，
我这块儿再加这个链接。
对，我已经看到了，我已经看到页面了。
已经看到页面，太好。哎，那也就是我不需要那个什么再重新输入这个。
哦，是吗？
对
它
对对，
它那个就直播页面不一样，我看它那个密钥和那个 URL 是一样的。嗯，
嗯，对对对对对，那太
但
好了。
是现在YouTube 就已经在直播了，我们先等一会儿再 share 这个 link。嗯
行
嗯
行，
嗯嗯，我看这个 link 只在我们那个小群里面发了，等那个
对，
开播之后再在大群里面再播，
好好好，
因为现在 YouTube 已经能够看到我们说什么。嗯。
嗯嗯，我看 Stephanie 也上线了，我把它设置为 promote to panelist。嗯，何博士还没有上线，然后 Richard 还没有上线。嗯，青在了已经，我把青 promote 为。嗯嗯
听我说话吗？我是 Richard。
嗨嗨
哎，
，Richard，因为您的名字没改，所以我看您举手了，我
哦，
就让你
对，好
对对对，没
，John
关系没
as a
关系。
panel。
能听见，Richard 你好，你打一下那个摄像头看看，你能看到你吗，Richard？
我把它改一下吧，我这块儿可以改。
能看到我吧？我打开摄像头了。
好，
看见了，
我把您
OK，
名字也改了，我
很好，
看一下。
很好。嗯，
现在就是
好，
河博这边没上线。嗯，
达克雷也上线了，Stephanie，Stephanie，
对，Stephanie 也上线了。
今天我们那个 speaker 也上线了，对吧？就
对
最后
对对，
那个，OK，那所
嗯，
有都上来了，太好了。
我们只有何博没有，何建博士没有上，就第二位 speaker，剩下的都上线了，都在线了。嗯
嗯，是永强跟他联系的，对吧？
对，那是一个大忙人，我前天在那个 LA 的时候还见他了，
啊，
催他，然后他把 slides 寄过来，然后对，他就
现在跟
是
他发个消息催他一下吧，要不？
可以啊。
我也来在群里，咱们可以上线了。
他最后1分钟还要调位置呢，他这个不太可能的事儿。
我们现在是已经在放人进来了，对吧？有 attendance。
因为今天是 webinar 的形式，所以就是把speaker 都设为 panelist。嗯。
Sure。 我们今天是所有的嘉宾都是在美国这边吗？都是晚上。
对，我开始以为何健博士是在国内呢。
没有，他在加州，
哦，对
他在加州。对，
对对对，
我们前天是那个，就是我们做那个ADLM，就是Diagnostic Laboratory Medicine 的那个 annual meeting，他也在上面，他也在里边。
OK，所以他在加州现在是他的6点钟。
对，
对，忘了青在上海，青你那边是上午。
对，我在
你看，
上海，Stephanie 应该是在中部。对，
刘刚刘博士跟我登的是一样的 background，我们俩就是一个镜像，都好像是在海边似的。
然后我可以换一个。
没有，这个挺好的啊，
对，没事。
挺好的，
开启
挺好的。
度假模式。
对呀，
我觉得我是在加州嘛，所以既然是在
挺好的。
南加，
哦，你
那个
也在加
对那
州，
个金陵
那我们有
大桥用的
2位
就不合
speaker 在
适了，
加州。
我就改成
挺好，
B 去了。
挺好的。
我们现在人数越来越多了，陆续上线了。
全可以实时的联系到那个我们的第二位 speaker 的。
那微信这边我就开播了。
可以再稍微等个2分钟。
那个 Dina，待会儿我们俩穿插的时候是说是你前面把这会长什么都弄完以后，然后 speaker，然后你一个我一个，然后你一个我一个，是这样的，对吧？
对对对，可以吗？我有
可以，
一个
对，
大概的这个顺序。
很好的。尤其你做那个 GPT 那个也挺棒的，把他们 background 以后都 summarize，省得我们自己去编。那 question 呢，倒是到最后。
我们的 question 是在这个25分钟之内的，就是我
对，
们后面就没有统一的 Q&A
对
环。
对对对
嗯，所以一会儿也会鼓励大家，就是在最后，就是每个 talk 20分钟，然后5分钟可
对，
以，可以分享自己的问题，也可以打在这个 box 里边，然后我们也都可以看到。Okay. We are having more people, more audience in already, so it's a good time to start. Good evening, everyone, and good morning to those who joining us from China. Welcome to our CBA 2026 Q2 quarterly highlights review. I'm Dina Du from AstraZeneca, and I will be your host today, alongside with my co-host, Dr. Yong Wang, President of One Day Henrico. We have an incredible packed agenda with 8 industry leaders tonight. To kick us off, I would like to invite Dr. Angela Zeng, CBA President and Global Clinical Lead at AstraZeneca, to deliver the opening remarks. Welcome, Dr. Zeng.
Thank you, Dina. Good morning, good evening, everyone. Depends on where you are. It's my great pleasure to kick off this quarter review. As many of you know, CBA Quarterly Review is a new brand started this year. So we do want to gather all the information together and share the information with you. More importantly, to have the discussion with you. As we know that it's so fast-paced in the biopharmaceutical industry area, so a lot of highlights, a lot of hot topics. But we know that from different perspectives, you might have different interpretation. So that's why gathering the peers together to have share the information, to have the debate, to have the active discussion, I think that's very important. It's also really helpful. So that's why we want to have this. We want to invite our speakers with intensive experience from their field, also to share their insights, also to have a discussion with all our attendees. I think it's a great time to have this the second time. We understand it's an evening in the US for those audience. This is a Friday evening, so many of them probably you need to spend time with family. And also for those folks joining us from China or from the other side of the globe, we know that it's also weekend for you already. So it's not easy, I think, to really spare the time to spend the time with the co-workers, with the peers in this field. But it's also a time really enjoyable because we have these common topics. So with that, I will pass here. I will just hand it over back to Adina. I think it's a great time to kick off all these topics. As you saw our posting before, this is another great session with 4 topics. I think I'm really looking forward to that. Back to you, Dina.
Thank you so much, Dr. Zhang. We will dive right into our first segment from our non-clinical groups. So it's my pleasure to introduce our first speaker, Dr. Xinrong Chen, toxicologist and founder of ABC Toxicology Consulting. With over 20 years of experience spanning the FDA, CPSC, EPA, academia, and industry, she brings unparalleled expertise. So tonight, she will decode the FDA new guidance for MABL. Dr. Chen, over to you.
Thank you, Dina. Yes, I will discuss the new guidance MABL, minimum anticipated biological effect level. Thank you. So you will control the slides. Yeah, the regulatory background for the US especially focus on the drug regulation for the FDA. So the drug regulation goes back to 1906. That is how the Pure Food and Drug Act gets started. And then in the 1937, there's a disaster caused by elixasulfonamide. More than 100 people died because of this accident. So the Congress passed the 1938 Food, Drug, and Cosmetic Act. That is the one we are still using today. So that is the law. And then the next level is the CFR, or Code of Federal Regulation. The important regulation for the toxicology or for non-clinical is the GLP regulation, which is the 21 CFR 50H. Next. The toxicology data requirements for the drug development. So as we all know, the drug development has 3 phase 1, 2, 3, and then before that there's the research and development. After that there's post-marketing monitoring. So toxicology are involved in every stage. Before the phase 1, there's the IND checkpoint. And after phase 3, there's the NDA checkpoint. For the IND submission, usually the sponsor will provide pharmacology, safety pharmacology, PK, PD, ADME, single dose toxicity study, also repeat dose toxicity study, which is usually the 4-week or 28-day animal study, and also the genotoxicity study. And then under the later stage, there is a long-term toxicity study, or the reproductive and the developmental toxicity study, which include FED, EFD, PBND, and for small molecules, they also need carcinogenicity study. Next. So we are talking about the MABEL guideline today. MABEL is about the dose selection. So before the MABEL, there's a nowhere approach. This is the standard approach. As I mentioned in the previous slides, the sponsor will do some toxicity study. Usually, through a good study, you will get a nowhere, which is the no observed adverse effect level from the toxicity study. After you have the nowhere value from the animal study, you can convert that to the human study. FDA has the guideline to show how to calculate. This slide show if you convert the animal data to human equivalent dose, and then the factor for the rats, I highlighted here, you divide a number 6.2, and then you get the human dose. Next. So an example show how to calculate the human equivalent dose and then the MRSD. So start with the animal. For example, here, the nowhere is 10 milligram per kilogram per day. That is a typical study. For example, if you start with 3 dose, 3, 10, 30, usually the mid dose, if the study design is very good, then the mid dose, 10 milligram per kilogram, it may become the nowhere. So with that nowhere, we divide the number 10 divided by 6, and then we get 1.6 milligram per kilogram. That is the HED, human equivalent dose. And then you apply a safety factor of 10, convert that number to the MRSD, maximum recommended start dose for the human. So that is the dose for the phase 1 clinical trial. Next. Now we will move to the MABEL. So there's some history. Why do we have the MABEL? It's basically just like the Food Drug Cosmetic Act. There's a big accident or incident or a disaster. Here it is the TGN 1412. This is a candidate drug. It's a monoclonal antibody. So they follow the standard pathway to conduct the animal study. Here it is the monkey study. They use the different dose, control 0.1, 0.5, 50, and with the highest tested dose, 50, there is no significant toxicity. So the nowhere is the highest dose of 50 milligram per kilogram. And the safety margin is very high, in this case, 500. And it's through the IV dosing. There are 2 monkeys in each group. The animal are treated for 7 days. There's no significant toxicity or other effect identified in this study. For the safety pharmacology, there's no adverse effect in cardiac toxicity, respiratory, or central nervous system. Next. Then they start with the human study. This is where the tragedy gets started. There are 6 volunteers in the phase 1. This is, I think it's in Germany or in UK, is a combination. And then there's a severe adverse effect because of the cytokine storm. And most of the people are hospitalized for several weeks. And they are multiple organ failure within hours. The effect is very, very quick. It's more like a acute toxicity effect. Next. And then they conducted some mode of action or mechanism study. They compare the human and the monkey for the cytokine study. So they choose the interleukin 2 and found the human and the monkey there's a big difference with a very low dose. There's a high expression for IL-2. But for the monkey, even with very high dose, here show 1,000 microgram per ml, there's no effect. So there's a big species difference for the human and the monkey. That means the monkey cannot predict the human. That's basically the reason why it's happening in human but not in the monkey. Next. So because of that, the TGN 1412, the incident was in 2006. And then there are lots of discussion. Also, EMA, the Europe similar agency like US FDA, they passed the MABEL guidance right after the event. So the guidance was released in 2007. The MABEL concept was the first accepted in regulatory through this guidance. And we can see I highlighted here. So MABEL is the first time mentioned in the regulatory guidance. Although there are lots of discussion before this guidance in research or in pharmaceutical preliminary work. Next one. So the highlights for the MABEL guidance. Basically, they indicate the toxicity, the traditional toxicity method or the novel method is not working for some of the biologic, especially for the molecular antibody. It needs data integration in addition to toxicology. The pharmacology, the PK/PD data, the target biology, and the species relevance data all need to integrate together in order to get to lower the risk. And again, the EMA MABEL guidance is the first one to recommend use MABEL in the regulatory history for biological such as monoclonal antibody, bispecific antibody, cytokine, and other novel biologics. Next. And then in just last month, in June 2026, see the clinical pharmacology division published the MABEL guidance. Similarly, they integrate all the data for pharmacology, toxicology, but they highlight the modeling approach. So in the title, the guidance of the title, they use QSP, quantitative system pharmacology. So there are also discussion for the modeling part. And also FDA highlights this is within the model-informed drug development framework. The other parts are very similar to the EMA guidance. Next. So just like any other in silico approach, here is PBPK modeling. So you need to validate the model. It needs to be scientifically sound and to be reproducible. And the model needs to be has good transparent. So the method needs to have a clear structure. The assumption parameter, data source, or software modeling rationale need to be clearly described so other people can review, can repeat, or understand the process. Also, the uncertainty and the sensitivity need to be assessed. Next one. So what the difference for the EMA guidance and the FDA guidance? So we got from the title. So EMA focuses on the risk reduction, risk mitigation, because it's right after the TCM 1412. But for the FDA, it's more focused on the modeling part. And it was published after the FDA roadmap. which is focused on the animal reduction or in vitro study or in silico study. Here it is in silico of the modern part. So they have different timing or background. And the title, FDA puts the label officially in the title, so we can call it the label guidance. But for EMA, it's very similar. The title similar to the FDA old guidance. The novel approach guidance is still called like the first in human or the dose selection, this kind of terminology. FDA is the first time to officially put the label in the guidance title. Next one. This is a case study for Mabel. So the drug is called glofilmab. It's a bispecific antibody. It's from Genentech. FDA approved the drug in 2023, so it's pretty recently. It involves the T cell activation. So the indication is for B cell lymphoma. For the therapy, it uses 3 different doses: 2.5 milligram, 10 milligram, 30 milligram. Next one. This is the FDA original document. I cited the original FDA language. Basically, they indicate the official, the MABEL first in human dose was calculated from the IL-6. They measured many other toxicity endpoints. IL-6 is just one of them. It's end up IL-6 is the most sensitive one. This is not my slide. Hey, Dina, are you there? Okay, thank you. The next one. Yeah. So the real method, there are more details in this publication. These are come from the company. They publish the paper in journals, so we can see them. The drug is bispecific, and it can connect the T cell and the B cell. The T cell marker is the CD3, and then the B cell marker is the CD20. And the calculated method based on the IL-6. So they first conducted the in vitro study, and then the IL-6 was selected, and then they used the PK modeling to calculate and get the MABEL dose to be 5 micrograms. That is very low. In the previous slides, I showed the therapeutic dose is 2.5, 10, and 30 milligrams. We can see here is 5 micrograms, almost 1,000 times lower. So it's very low. And then they did in the phase 1 and phase 2, they conducted a typical dose escalation. Each time they increase about threefold, from 5 to 15, 45, and then 135, and then every time increase about threefold. Yeah, finally come to the age 10, and then if you multiply 3 again, become about 2.5. That is the therapeutic dose. Okay, next one. Finally, this is my last slides, or the scientific slides. So similar to the TGN 1412, recently there are lots of discussion for this is also kind of disaster. So one it's a special special case, it's not go through the IND, it is through the IIT, or is another pathway in China. Also, they have this pathway in the US, but in China, looks the regulation is not very stringent. So in this case, they have expert committee reviewed the study. Some of the experts raised the question, but unfortunately, they didn't stop the study. So the patient, which is age, no, 6 years old Chinese girl, died only in 7 days. And then there are lots of discussion for this tragedy. It's people are talking about the dose, but in this case, it's less about dose. It's more about ethical issue because the people are not following exactly all the regulation or the guidance or all the procedures, and then especially for the transparency, people have concerns. So that is kind of the major discussion people are talking about. Next one. Oh yeah, that is the end of my presentation. If you have any question, I'm happy to answer. Or if you have any comments, I would be happy to hear your comments.
Any questions from audience? Adida, do you have other questions sent to you?
I haven't seen any questions here from what I can see here,
Okay.
but please feel free to leave any questions in the chat box because we have 5 minutes Q&A. And if we couldn't answer your questions in time, we will follow up.
Okay, great. I think it's thank you so much, Dr.
I
Chen.
do see, sorry, I do see there is a
Yeah, go ahead.
question. People are asking, could you clarify the difference in MABEL between Europe and the US again?
The data application are pretty much very similar. Because you have to use all the data for any kind of analysis. It doesn't matter if it's novel or Maple, you have to use all the available data. So including the pharmacology, the safety pharmacology, the PK data, the PD data, and the toxicology data are all used in the EMA and in the FDA guidance. But for the FDA guidance, the guidance itself, when you read the guidance, there are many pages talk about the modeling. So there's some progress with the PKPD modeling after 20 years of development. I think that is a major difference. For the endpoint itself, there's no big difference. It's all for biological, especially for monoclonal antibody or for cytokine involved study. So I would say that the timing is different. So they are more modeling approach or criteria in the FDA guideline.
Thank you. There's another question mentioning so you mentioned you highlighted the shift from the 2007 EMA MABEL concept to the recent June 2026 FDA guidance emphasizing the QSP models. So what do you see as the biggest operational hurdle for companies trying to integrate these QSP models effectively to satisfy the FDA's new expectations?
I don't know if the FDA a new expectation. So it depends on the molecule. Actually, if it is especially this is about immunology, biology. If your molecule or if your drug is a biological and is involve cytokine syndrome or cytokine storm. And then FDA, at the IND stage, they will recommend, or FDA may ask the question, if you use, for example, I see in the case if people use the NOVA, because NOVA, the dose is higher, then the safety margin will be much higher. But if you go with the MABEL, since the dose is much lower, especially for the clinical trial, the dose escalation, there are much more work to do. Of course, the cost, the timing will be take longer, but it depends on the safety or the molecule itself. If it is a T cell or myeloid or immune cell involved, and then you have to consider the MABEL method. It's I would not say it's not a new requirement. It's based on the context or the molecule itself.
Okay, thank you. Thank you so much, Dr. Chen.
Thank you.
Yeah, I think that probably is that we move to the next speaker. Thank you so much, Dr. Chen. This is very important, new guidance from the FDA. That's a very new, and that start from in June. So the next we move to the clinical translational medicine group, which I and Yixing lead that group. We are honored to have Dr. Jian He. He is the president of R&D Health. Dr. He is a physician by training and also is an entrepreneur. And he is the pioneer in functional medicine, the longevity and digital health innovation. He has a lot of more experience in the translational medicine and then the big health and so on and so forth. Today, he will be tackling a massive topic. For the title is the $1 trillion question for translational medicine. Translation to what? Here you go, Dr. It's over to you.
OK，谢谢永强兄。你们是放片的还是我来共享啊
Dr. Hu, please share your slides. I will stop sharing. Thank you.
OK, so I can share my slides. Hold on. Make sure that I got my slides right. Oh, I have to send a request for sharing the slides.
Yeah, I have approved.
Oh, okay.
Yeah, so please share. Thank you.
Dina, also you have backup slides, right?
OK，
Yeah, we can see.
可以看到吗
Yeah, 可以。
？OK, great.
Yeah, you can speak your English pretty good. I think it's since we may have some the speaker and who don't understand Chinese. Just on the
Oh, really? Oh, you got someone that don't understand Chinese? I saw this is a Chinese association.
yeah, it is, but it's on the English is the official language.
Official language. Okay, great.
All right.
Well, maybe I should just briefly introduce why I like to participate in today's activity. Yongqiang兄呢, we know each other for over almost 20 years, and I understand that he's very active involved in many associations. This is one of them that really impressed me because he was telling me that there's a lot of scientists that's really working this field, and you have a small group, 就是一个小组, right? I made a slides in English, but I translated into Chinese, so that make it difficult for me to do entirely in English. Okay, so it's not that I have to, I want to, but now it seems that I have to speak Chinese. Okay, well, the future translational medicine, as we see, is changing. Okay, and I'm going to talk a lot about why it's changing and what they are changing to, and how much that we may be able to be prepared, and why I am thinking about this way, and what kind of work that I've done that lead me to talk about this topic. Okay. Um, well, if you look at, I'm sure you read Chinese, right? 永强咱们这里读看中文没有问题吧？Right?
Dr.
Okay.
Hall, this is we have the live stream broadcasting, so I think it's better to stay in English, please. This is Dr. An
Oh, okay.
Xuanzeng. Yeah.
Oh,
Yeah,
okay.
we have
Good.
the multimedia, you are just
Yeah,
saying that
yeah, we have
in
YouTube
the
or the WeChat, so I think
YouTube,
it's better.
Zoom,
Yeah.
and everywhere now, real time.
Okay. Very good. Very
It's
good.
also
Okay.
recording, so please. Thank you. Go ahead.
Okay. Fine. Okay. I'll go on to why I call it a $1 trillion question, because it's big. Okay. And why we're asking translate to what? Because we are really trying to figure out what is the targets, what we're trying to convert something in the laboratory into the clinical applications or into the final application. So here, what I'm asking here is what's the endpoint? Well, in general, that when we do the translational medicine, we're producing a product. Okay, so this is the traditional classic approach to do translational medicine, and we know it's still going on, and there are lots of pipeline products being produced. That's okay. But what I am talking about is what's new. Okay, well, the old way to do things that I call it, there's a lot of waste. Okay, the reason I said this is it has to do with my background in laboratory medicine. I was trained at Mayo Clinic, and in laboratory medicine, I've been doing laboratory medicine throughout my career, and I have still have laboratories that doing clinical testing. So we know there are lots of data being generated from the lab. And 2 years ago, I translated a book. It's called Patient Equation. The book was written by a very well-known guy. He's the founder and CEO of Medidata. So he has accumulated a lots of data, and he had a very good experience in looking at the data that's not being used, literally being wasted. For that reason, he wrote a book, and he clearly indicated that there are lots of data. The reason we cannot use them because we don't know what it means. There's no way to know what it means. We're collecting those data, but the data is being stored somewhere, never ever being used or put in a use. Now, when I call it, it's a waste because we have the opportunity to collect those data. I'm going to throw my talk. I'm going to talk about data, data, and data because there's so much data that's so important that we have to make a good use of it. And that's actually going to help you to develop much better products, or you can help to get through all kind of hurdles. Okay, I'm going to talk a lot about that. So definitely we need to break the boundaries, and we need to have a disruptive thinking process. When we're talking about disruptive, meaning that we probably have to overcome a lot of our own personal thinking, our own experience. Now, the first disruptive thing that I believe is, as being practiced already, is that instead of doing laboratory experiments I also have a PhD degree. I remember the day that I was doing my PhD work. I have to do lab work every day and try to make it work. I take a lot of time, waste a lot of my graduate school advisors' money, grant money, um, and we generate a paper. That's all we do. Now, today we have seen so many drug discoveries are not being done by or in the laboratory. It's done in an office on a computer. So this is, I don't have to talk too much about it, but when I search, I have some data here, I think it's very interesting. When I search, there's a list of things that AI or computing can help for drug discovery. Okay, starting from identifying targets, doing like protein structuring, um, even generate brand new molecules. Okay, there's a list of it. When I look at there's a review from FDA. This review is pretty interesting. They actually review since 2016. There are over 500 submissions of whatever clinical trials. They count everything that's submitted to FDA with AI computing. Okay, since 2016. Now, why? From what I see, this is pretty early, and so many people already involved in AI and data calculations, that kind of stuff. So it's not new, but I believe people have not taken it entire, starting from the beginning with calculation first. I don't think it's that popular yet. I mean, 500 is not much less than what, how many people are doing drug discovery, in this country. So we probably have to think back that let's do calculation first. Let's think about what we can do before we go into the laboratory. Now, the second one is this is fascinating. I have done a lot of animal work even since my medical school time. We use mice and we use different kind of animals. So to develop an animal model is quite routine for any kind of drug development. Now, the question is what we're getting from the animal model. In fact, most of animal model only serve a single purpose. Well, the reason is we already have whatever target molecule or target mechanism that we want to prove in the animal model. Now, today, we see any drug when they go into the human body, and not just go into target whatever mechanism that we discover from animal model. Because human body, we have a lots of interactions with your own physiological status, with diet, with environment, now even with your mental health, right? Your emotions. So what I believe, and this is actually also happening, the animal model now is not just doing serve for a single purpose. They're going to collect all the data for this animal. Okay, you have to collect the diet, environment. Maybe you have to do sort of like a digital twin of the animal and make sure that you understand this animal much better than just a single mechanism that you already target for. Okay, so later I'm going to talk about, well, actually, the second point is exactly what I mean, is that when you collect all these animal models data, that you're going to correlate that with human model. Okay, that's very, very critical because if you don't understand the correlation between animal model and human model, you probably don't want to collect those data. Like the digestive tract of the animal compared to humans. Okay, what kind of enzyme involved? What kind of microbiome involved in these drugs, in this animal? Okay, so we need to collect a lot of data, including microbiome of the animal, and then we correlate that with human models. So that's what I mean by another disruptive approach that's coming is rebuild a animal model. Now the third one, this is even to me, it's a given. right? I mean, we are now living in a internet era. We know internet. We know the whole body is the internet. We know human beings who interact with the whole world, that's the internet. So when we do drug discovery, how can we just think about one target? How can we just think about a mechanism? How are we still thinking in a linear approach? So I list a bunch of things here. I mean, I translated into Chinese. Actually, I'd rather to have everything in English so you guys can read it, right? When we're talking about genomics, we're talking about epigenetics, that's a lot of data. That's by definition is part of the internet that we're talking about. We're talking about metabolism. We're talking about absorption. Oh, by the way, I listed here, liver is an organ that does a lot of things for the body. I mean, we understand they do metabolism of drugs. But what we don't understand is liver is also help the body to manufacture a lot of molecules. Okay, I mean the most known one that we know is cholesterol. I mean LDL is manufactured in liver. So and they also help us to clean up our body waste. Okay, therefore understand liver status, the liver function, how well the metabolism pathway in the liver is so important for drug discovery because most of drugs still metabolize in the liver, unfortunately. Okay, so liver is tired. Okay, if you load up with uh with the waste with well some of them not waste, some of our good stuff that need liver to process, the liver will be fed up. Once they get fed up, then there's a problem. It's like you go to like from where I live, going to LA downtown. Um, you constantly we end up in a huge traffic jam on a highway. Okay, and that's exactly what's happening in the liver when you don't understand how well those metabolism channel, how well the enzyme system, and we know P450, right? I mean, we can do genetic testing and figure out if you have any kind of defect genetically. However, there are lots of your lifestyle, your environmental hazards around you has to do with your liver, okay? And then microbiome, and then lifestyle, and then well, I think it's very important is the time scale, okay? Human beings live in a very interesting time scale, like when our body knows, our biological clock knows when you go to sleep, when you have a better, more intelligent during the day than other times, well, sometime in other times. I'm actually another specialty that I'm involved in is functional medicine, and we have a test from saliva. We test your cortisol level, and your cortisol level is pretty high in the morning and go very low in the evening. And cortisol has to do with your energy. And when you say you do some more intelligent work, you need more energy in your brain. If you don't have enough cortisol, your energy level is going to be low, and that's not the best time for you to do more creative work, or you do negotiations with your business partner or your collaborator, because your brain not function as well. So understand um, your biological clock and also understand uh, in a time screen on a daily basis or on a weekly basis, understand how well that your body are in a better status that has to do with your health, has to do with your lifestyle, has to do with a lot of things. So in general, we are living in the internet era. When we talk about drug discovery, we need to think about the body as the internet. Okay. There are a lot more we can talk about. Oh, this is actually my uh, invention in about 10 years ago. This behind in my laboratory work for many years, and we are doing a lot of work on that. Actually, I'm a professor in Peking Union Medical College. I have my graduate students. We did a lot of studies on one of these. Okay, so when I look at human body, I I see it's a omics. Okay, we all understand what omics is. So I uh, raised a theory on hexaomics, so starting from functional genomics, functional macronomics, functional microbiome, functional structuromics, and functional metabolomics. And the last one is functional lidomics. Literally, it's lifestyle, environment, and diet. And there are lots of data, and we're actually building quite a few models around this theory. Very, very interesting. If people are interested in this, we can share this later. Now the fourth one is we will do a digital twin. And this is a term that's very, very popular in today's digital world. Okay, we're all talking about digital twin, but in drug discovery, in translational medicine, we need to really put this in use. Okay, we need to generate a real digital twin for any kind of studies that you're doing. I hope that people, everybody, when you do translational medicine, you can't put digital twin lightly. Okay, this is very, very important. I don't want to read everything on my slides. Um, Now, I'm sure you can read this. I think translational medicine need to translate yourself first. Okay, we are doing translational medicine, and we know we have rules, we have all kinds of process we need to follow. However, we also have to understand that in order to I guess in order to survive, maybe this is too heavy. In the future for translational medicine, we need to translate ourselves. And here is the reason. Okay, clinical studies is not the final exam. Okay, well, you may say it is because once we get it done, we get a product out, we get FDA approved. If it's medical equipment or it's the drugs, that's it. But what I am telling you, the future of translational medicine is this. You have to build a system that will continue learning. Learning what? Learning how well your products being used in the market. Learning the data that you're collecting through your products, and that will help you to optimize, make your products better used. There are a lot more that we need to learn. So clinical studies has to be continued for, I wouldn't say forever, but for a long time. Okay, another new things I believe is we can always use a marker, two marker, three marker. We have to consider the entire body. We have to think about the status of the body. Okay, in order to do that, we have to collect not just laboratory data, well, clinical laboratory data. We have to collect imaging data. There are lots of omics data. There are variables. There are lots of variable device, like a ring, like a watch. You collect continuous data, and you have to use questionnaire to collect patients, their own feeling. Okay, their symptom changes, their lifestyles, etc. So we need to collect a lot of data versus a few markers that we are using today. Now, why this is possible today? Well, if you collect so many data, the book that I mentioned, Patient Equation, that I translated into Chinese, that the author said there are so many data that collected that people don't know how to use them because there's no tools to analyze the data. It's not like that we don't want to use them. Sometimes human beings want to, we have a feeling, we think these data may be related to whatever, but we have no tools to analyze them. So today, AI models will help you doing that. Okay. So what I'm saying here is when we have a opportunity to collect the data, we just do it. I mean, we can simply leave all the data to AI. I don't know if you all heard of, sorry, I only have a few, okay, 5 minutes. There are quite a few. There's a very well-known story. Is a guy, I forgot what company founder in Silicon Valley. He apparently he got cancer, and the doctor said that you probably only have 3 months to live, and um, we don't know what to do. We can save your life given the cancer that you have is pretty rare, very difficult to do any kind of treatment anymore. And this guy, he said, well, if that's the case, if the doctor can't do nothing to me, I'll do it myself, even I'm not a doctor. So he did all the tests that he can do in the whole country. He go to this laboratory, collect, CAT scans, MRI, all these data, and he put them together. And the doctor said, are you crazy? I mean, nobody knows what these data means. Why you collect them? He just loaded the data, 6 GB, well, 6 TB, whatever. It's a huge amount of data into a computer model and let the AI model to handle those data. And we all know the story. This is the big news. At the end, the model give them that there are a pharmaceutical molecule, whatever, that can treat this type of cancer. And then he went down, find it, and have a whatever pharmaceutical company in Germany, and they actually produce a drug for him. After 3 years, he's still alive. Okay, so what that tells us? That tells us that collecting data, enough data that even you don't know what it means, they actually mean something. Well, longevity will encourage, or well, longevity require translational medicine to change yourself, because today we see people getting older, we see people getting to 70, 80 years old, 90 years old. They're not 28 years old, they're not 18 years old. 18 years old, we call them adult, but when they are 88, you still call them adult. So that's not fair. In medicine, that's not fair, right? We know they're not the same. So when you do translational medicine, you need to consider longevity. There are a lot more that we can talk about. Functional medicine will help translational medicine to change. Why? Because functional medicine, by definition, is a integrative, holistic approach to analyze the body function. This is the article that I introduced functional medicine to China back in 2007. This is well considered as the first article that introduced functional medicine. And in the middle, this is the tree of functional medicine that tells how we understand human beings, chronic illnesses, functional decay, all that kind of thing. So I can talk, days about functional medicine, but what I'm saying is functional medicine is a series, a way of thinking, and that will help translational medicine to change. Well, new type of translational medicine is here. Okay. So I believe that we should go into a 5P, or you call it P5, from predict, personalize, prove, prevent, and finally is predict. So, and then at the end, I propose that 6 layers of a structure for translational medicine. This is pretty complicated. I probably run out of time to talk about the details. Well, by the way, the slides already in your organizer's hand, so they can share with you. The researcher, well, even with AI, you're not going to lose your job as long as you change yourself. If you don't, you may lose your job. So, well, looking at 5 years from now, what we should do today? Well, if you look at what I have here is data, data, and data. Don't forget that you rely on data, and data will help you a lot. Data is your friend, and as long as you treat them well, okay? Don't think that data, too much data that bothers you. It won't, okay? It will help you. There are a lot more that I can talk about if we want to talk about data. Well, finally, I guess the question's been answered. The trillion-dollar question is translate to what? To improve human health, to make sure that people live longer with a better health span. That's what translational medicine should do. Well, this is the end of my talk. Thank you so much.
Thank you so much, Dr. He. That's impressive. So if you have any questions, please leave your questions in the chat, then we will follow up. Considering of time, let's move on to next one. Next speaker. I'm thrilled to introduce our clinical regulatory affair group lead, Richard Zhang. Richard is also a global regulatory affairs director at AstraZeneca. He has over 25 years of experience leading global regulatory strategy and has supported over 15 NDA and BLA submissions globally. He will be discussing a major regulatory shift. One pivotal trial as the new FDA default is the two-trial convention over. Richard, the screen is yours, please.
Okay, thank you. Thank you, Dina. Hello, everyone. So it's my pleasure to be here to introduce the regulatory topic. I think maybe you're familiar with the regulatory function. This is one of the function part of R&D, and really can provide what the data we provide we generated and to share with FDA, submitted to health authority and get approval. This is general rule, but regulatory really part of R&D work and really can guide the drug development, especially for drug development. For today's topic, I will talk about the one pivotal trial. I think this is very new to the industry, but for it's a new signal, or I think it's a information shared from the FDA, previous commissioner, etc. And also we have updated the new guidance, 2024 guidance about demonstrated substantial evidence of effectiveness. So today I will introduce what's the background and what's the impact for our R&D work. So can we move to the next slide? Thank you. So today's agenda, I will cover 5 parts. The first one, I will talk about the FDA leadership statement. The previous FDA leadership team statement, this published on the New England Journal of Medicine. So the second one, I will show the history behind this. And third, I will compare about the guidance 2023 and 2026. And then I will go through a case study, recent example of this one trial requirements model recently approved by FDA. And the last, I will highlight the main takeaway for the R&D work. So the next slide, please. So first, let's take a look about the article published by FDA leadership team. Next slides. So I think this is the key message from this slide is that FDA is moving toward to adequate and well-controlled study, plus confirmatory evidence as a default for the product approval. This is not related new to the industry. Actually, FDA already had this option since 1997. I will show later in the history about this one. Also, it's a give a give a signal, give a information that what's the better trial design, and because the technology has developed, and also a better understanding of the disease, and also the biomarkers. So that's why, based on all this information, the second large trial may not always added much value for the drug development. So next slide, please. So I think there are 5 reasons for the ending the 2 pivotal study requirements. I think in the statement, but I think the previous slides already covered 1, 2, 3, the 1999 guidance regulations, and also the modern drug development, and also the industry. From the industry part, I think there are 60% of first approval recent 5 years really based on a single pivotal trial. And the next one, the fourth reason is the ethical. I think if if the trial already show the one trial or there's a very clear meaningful benefit, it may not really very difficult to justify repeating it again with a second trial, pivotal trial. And the last reason is innovation and access. I think reducing the duplicated trial really can reduce the development costs, and we can gain the fast approval and also a quicker access for the patient. Patient really can benefit about this one. So this is a final reason we see this, we need to ending the two trial requirements. Next slide, please. So let's take a look at the history of evolution about the regulation, about the requirements. Next slide. Thank you. Yeah. So I think it starts with it's a Kefauver-Harris amendment. I think this is 1962, go back, showing the effectiveness become a formal requirements. And at that time, FDA expected 2 independent trial, then followed by FDA Modernization Act. I think this is issued in 1997. That the one adequate well-controlled trial with confirmatory evidence could be accepted by FDA. This is a regulation issued. And then after that, FDA become more flexible, but it's still very, very low moving forward until the 2019. The draft guidance really talk more about the evidence and the trial design, but still kept 2 trials requirements at that time. In September 2023, the FDA issued a draft guidance focused on the 1 trial plus 7 types of confirmatory evidence. However, this approach was still treated as an exception for the single trial requirements. Until this June 2026, the revised draft guidance issued by the FDA really made the shift much clearer. They say the 1 pivotal trial plus confirmatory evidence was present as the main path. So this change did not happen overnight. And the FDA has been moving this direction for years. So let's move to the next slide, please. So let's compare the old and the new guidance more directly. Next slide, please. So I think here is a clearly compare side by side about the 2023, 2026 guidance. I don't want to go through the detail. I think the key change here showed, like in 2023, the 2 trials still be a default, but 2026, 1 trial plus confirmatory evidence is presented the main path. So the next slide show the regulatory approval path before and after. Okay, go to the next slide. Thank you. Yeah. So at the top is the old path. You can see the early development, normally phase 1, phase 2, and then followed by 2 pivotal study. Could be sequence, could be parallel to pivotal study, and then gain the approval if the data really can show the efficacy and safety and benefit is better than the risk. So the second trial usually expected. This is a routine required before. So the second line, you see the new path, the early phase could provide confirmatory evidence. Then the once drawn pivotal trial plus confirmatory evidence and can getting approval. So this is really a new approach, and this is what the FDA presented in the article and also in the new guidance. So the key message about the two pathway that the 2 trial is no longer as a default. However, the second trial may still be needed if 1 trial is not enough. For example, if the patient group is too narrow, the safety data are limited, or the study has important design issue. So the major change, summary in the next slides. Yeah, go to the next slide. Thank you. So the big change is this is very straightforward. It's a pivotal plus confirmatory evidence analysis default, and the guidelines also allow more flexibility, especially for serious and rare disease. I think for oncology and rare disease is very, very common for one pivotal study, but I think for other disease, I think really need to think about the change. And also the new guidance also can can include new method. For example, the Bayesian boring and for the biometric method really um, can discuss with FDA. I see FDA also encourage industry to have a communication with FDA expected to issue a Bayesian boring guidance in the future. So the key message from this slide, I think the one trial, it may be enough, but I think it's need a very strong trial. Means the p-value, etc., and the power really need to satisfy FDA. So I think this is really need a high authority interaction before you move forward with the late phase for the phase 3 study. So maybe can next one. I will go through a case study. And for nerandomilast, this is recently approval by the FDA. I think this product was originally developed by Bringer Ingraham, and it was approval by October 2025, last October. And for the patients with idiopathic pulmonary fibrosis, we call it IPF. The product was granted breakthrough therapy designation based on the positive phase 2A results, and it also received the priority review after submitted NDA. So the IPF, the disease is really very serious, and there were still a clear unmet medical needs. Even there already are two product approval already marketed in the different in the major market like the US, EU, and the China, etc. So let's look at FDA review process. All the data here, all the data I generated from FDA website was published. So during the FDA review, FDA first take a look what's the requirements they publish, and they will check the box. So very clearly they check. So for one requirements, two or more adequate well clinical control clinical investigations, but they say they check it qualify because in practice the sponsor submitted the two major trial. One is a pivotal trial, is a study 14 as a phase 3, and plus another study 13. This is a phase 2A trial. This is a proof concept study. But however, this study really can generate the efficacy data, can provide confirmatory evidence to support the NDA's approval. Can you go to the next slide? It's more clear about the study design. So look at here, the phase 3 study, the study 13, the phase 2A study, sorry, phase 2A is the first study 13, the phase 2A study. The treatment period in the 12 weeks, and the patients is 147 patients. Look at here, sample size not very big because it's a proof concept study, but it show a significant reduction in the FVC as an endpoint. FVC is a forced vital capacity. This is the endpoint to measure lung function. So this compare with the placebo, really can see the significant reduction. And the study 14 is a pivotal phase 3 study. The sample size is more than 1,000 patients, and treatment period much longer, is 52 weeks. So it also the same endpoint, FVC, and same endpoint, and so significant reduction. So I think based on the two study results, like the phase 2A study 13 provide confirmatory evidence, and the study 14 as the pivotal trial. So next slide, please. This is the very clear comparison, like the two trial as confirmatory evidence in the early phase developed generated data, and also plus the phase 3 pivotal trial gain the FDA approval. So this is really nerandomilast show the example that the FDA new guidance that one strong pivotal trial supported by early phase confirmed evidence really can gain the FDA approval. So go to the next slide. Okay, next one. So from this slide, from what we can learn from the guidance, also from the example. First, I think I will give the suggestion to the industry. Like first, we should stop assumption that that every product need a two pivotal trial. So the second one, when we design the early phase clinical study, think about the phase 2A study or phase 2B study. You know, this study really can provide confirmatory evidence in the early phase. And the third one, I think, if you want to really rely on one pivotal trial, the trial really need to be very, very strong. The data, especially you need to discuss with your statistician, you know, have the p-value really maybe, you know, 0.001, and you really need to have a very, very, very strong data to be shown to the FDA. And finally, I think this approach really need early FDA interaction, and we needed. I would strongly recommend it to have a FDA meeting, and for example, end of phase 1 meeting, even pre-IND meeting, at least at the end of phase 2 meeting, we need to discuss with FDA and to have a confirmatory evidence plan from the beginning. So if we want to one trial pathway, we need to design it very earlier. So the next slide, please. So this is my last slide. I think the closing summary. First, I think FDA is moving toward pivotal trial plus confirmatory evidence as a new default. And I think secondly, it does not mean the bar is lower in FDA. However, it means the one trial need to be very, very strong, and the supporting evidence need to be planned earlier. And the third one is the FDA alignment. I think the interaction with FDA really need to be planned earlier, pre-IND and phase 2 at least, you know, to be earlier to have the confirmatory evidence be planned. Some confirmatory evidence could be non-clinical data. Really, it's a case by case. You need to discuss with FDA earlier. And finally, the guidance, 2022 guidance is still a draft, so it is something we need to keep on eyes to see what's the next step. So in summary, this is not just about doing less trial, but it's about building a strong evidence package around one trial, one strong pivotal trial. And this is my last slide. Thank you. I'm happy to answer any questions.
Thank you so much, Richard. I think really a nice job representing CBA Clinical Regulatory Affairs to give this wonderful talk. And after you clearly mapped out the June 2026 guidance reverses the burden of justification. So there is actually a question on our live stream platform, Bell Valley. The question is about now that Dr. Marty Makary and Prasad have left.
Yes.
How likely will the incoming FDA leadership revert to the previous framework?
Yeah, this is a very, very good question.
Yeah.
Yes. I think the two leaders already left, but they they give the position. I think the statement is on behalf of FDA. So it's not the personal idea. And you can see the 2026 June guidance really follow up with later article. I think this is really, I can see where I'm very positive for the future for FDA to issue this guidance. They're still collecting comments. I think the industry really can contribute. What's your thought and what's your opinion? Really can make the change. So be, I think, be aware the FDA regulations, the guidance really need to be driven by the industry because industry really have the technology. We drive the direction. What's the next step? What's the gene therapy? What's the cell therapy? You know, the FDA never know if only the industry to move forward to the next chapter. So FDA need to catch up. This is what the FDA need to do. They did very good for oncology and the rare disease, but they really need to catch up with industry. I think I can see the I have been working many years in China. I see FDA, the China Health Authority really improve a lot, but FDA always the leading health authority in the world. But I think, but very interesting part, FDA really see the catching up from China. They feel a little bit nervous because China already improved a lot. For one example, they issue the IND new pilot. They can grant IND with 30 days, working days. And I see FDA issued a new pilot, very similar like what China Health Authority, the I N, NMPA did. They
Okay.
issue a new pilot and to almost copy China to say we need to improve our IND approval. We will combine with how to say they call we call EC. They call how to say the very similar. They need to ethnic committee to ethical committee to get approval involved in the early review part, and also the site need to involve earlier, and they really can not only approve, not only a grant IND to be open, but really can expedite the recruitment. So they are target recruitment as quick as possible. This is what they did. The China did the first and followed by EMA and now FDA also catching up. So you can see FDA are leading, but China is catching up. Sometimes some part of China is leading and FDA is catching up. They're very interesting. So
That's a great. I think so. I did it on a bit. The things take this opportunity to ask Richard about the global regulatory, the operation. What do you think that in the GCT area, the
geez.
gene and the cellular therapy?
Oh, yeah. I'm not really working with genes and the cell therapy, but I see the FDA is very eager to involve in the review. I think I just got an information that a new product with approval is from Regeneron. It's for hearing loss. It's called otamine. I maybe my pronounce me not accurate. So I think I have the information I just collected it. This is gene therapy and
Mm-hmm.
FDA approval only within 61 days. Can you imagine?
Mm-hmm.
And this is a BLA filing, and the FDA grant how to say fast track and regenerated medicine advanced therapy. They call RMAT. This is really new to me, but they also gain FDA accelerated approval. This is not a formal approval. This is only accelerated approval. And their study very interesting. The clinical study only single arm study, and they compare with history background, and only 24 pediatric patients from 10 months to 6 years, 16 years. This is adult to 60-year adolescence with confirmatory evidence, including the mechanism and from non-clinical data. So you can see this is a gene therapy. Really, they move even further. They are leading the industry. We hope if we really can use an AI anticipate and the drug work from animal and to the human, we really need to do a nice clinical trial and really can develop quickly and gain approval very, very quick. The patient can access the drug very easy and faster.
Thank you. It's a very good comment.
Thank you. My pleasure.
Is there any other questions from people online? If not, I think that'll be good. So that we move on forward. Thank you so much, Richard. He just summarized the recent happening, and then based on his on the own experience, and so for the case studies. For our final core segment from the CBA Frontier Insights Group, we have Stephanie Lee, the Director of Global R&D Sourcing at B1 Medicines. Stephen brings a wealth of expertise in healthcare consulting, M&A, and growth strategy. And people may be familiar with Stephanie from the CBA, but for this time we get larger about her. And then Stephanie, she will guide us through behind the GLP-1 gold rush. That's at Lilly's strategic pivot and the new capital wave. She already shared some of the information with us in the past, and now for the second quarter review, we're glad to have Stephanie. The floor is yours.
Thank you. Thank you so much. Good evening and good morning, everyone. I want to first thank CBA for the opportunity to share the Eli Lilly story at this quarterly review meeting. Today, I will walk through the timely case study of Eli Lilly, what happens after their GLP success story. This offers an example of how a major biopharma company can convert their product momentum into strategic capital deployment, external innovation access, and long-term competitive advantage. The core question that I want us to consider today is, when a company suddenly has extraordinary cash generation from a breakthrough franchise, how does it decide where to invest? And what does that mean to innovators in China and globally? Can we move to the next page, please? Thank you. As we all know, GLP-1 has become one of the most important growth engine in global biopharmas. So as expected, when we compare Lilly against their other large pharma peers, we see that if we just look at their Q1 2026 performance, they all performs across all different metrics. It is very clear. So their cash engine was supercharged by the massive 56% year-over-year revenue explosion to 19.8 billion in this first quarter. They're actually going to announce their Q2 performance next week. So I'm excited to see what happens next. But their first quarter success is really produced by Mounjaro and Zepbound, this producing that extraordinary growth and free cash flow, which give Lilly a level of strategic freedom compared to many other peers that they do not have. If we look at Merck, they are actually preparing the Keytruda patent cliff. And for Pfizer, they are managing a heavy debt burden following the Seagen acquisition and post-COVID normalization. for their competitor, Novo Nordisk, they remain strong, but rather than aggressively hunting for outside biotech platform in Q1, they actually funneled their capital into expanding its existing manufacturing footprint, just because they want to scaling up the production for the core products on Ozempic and Wegovy to reverse the recent market share loss to Lilly. So we can see for Novo Nordisk, they actually have 4% decrease in the revenue. So why this is important for our discussion today? So we see that the financial comparison really matters because capital capacity directly shapes the strategic behavior. We can see that Lilly in this first quarter, they actually behave more of like offensive pursuit of expanding their margin, strong cash generation, and then try to expand into other strategic advantage areas. Okay. Let's move to the next page. So I'm going to spend a little bit more time on this page. So far for this year, Lilly has been on an acquisition spree and completed a total of 16 deals across 6 therapeutic areas, which total of about 47.4 billion value. So their portfolio is broad. It ranges from oncology, neuroscience, genetic medicine, immunology, infectious disease, and then metabolic disease. So I want to say that the mapping here is a rough mapping because some of the deals actually can be mapped to multiple rows. But again, this is not unfocused. As you can see here, the top one focus area for Lilly is oncology. So a lot of their major investment is in this platform, like in vivo CAR T, ADC, multispecifics, and targeted inhibitors. And I also want to talk about the 2 of their largest acquisition this year. One is Colonia Therapeutics. It is one of their biggest M&A deal with $7 billion focusing on advancing their in vivo CAR T therapeutics. And then next to it, we have their largest Innovent licensing deal, Innovent Biologics. That's if we say it in Chinese, that's 信达生物. I do want to note that this one is not a M&A, it's a licensing deal. So for Lilly, they are partnering with Innovent to have the end-to-end Langes partnership. They are leveraging their Innovent's R&D and clinical execution in China to co-develop and commercializing high-value oncology, immunology, metabolic therapies globally. So for this audience, I would want to highlight that this partnership with 信达生物, it shows how China origin company can become a global validation and licensing partner, not just a lower cost development site, but a source of globally relevant innovation. I think this is an important shift in how multinational companies view Chinese biopharma assets. And then if we move down to the next row in the neuroscience, no, sorry, stay on this page. Next row, the neuroscience, the Lilly is actually moving into orexin biology, 食欲素, and the new modalities for depression. In this one, we want to highlight two acquisition or licensing as well. One is Centessa Pharmaceuticals. It's a major neuroscience move targeting sleep-wake disorder like narcolepsy, 嗜睡症. This is an expansion of their neuroscience pipeline. And then they also have acquisition with 4E Therapeutics. It is a bet on next generation non-opioid treatment for chronic pain. And then for genetic medicine AI, they actually partnering with many AI companies trying to change the discovery and development model. A few key ones we want to highlight here. One is in silico medicine. They want to use in silico AI engine to accelerate and drug discovery and development of those novel therapeutic across different therapeutic areas. And also there is that Ona Therapeutics. This one will bring in next-gen therapeutic technology, including in vivo CAR T cell. And then we also see that Eli Lilly is aggressively expanding its immunology footprint to novel platform like oral anti-inflammatory and to drive long-term immune reset and systematic disease control. And then lastly, we also see that after exiting the infectious disease category for 7 years, they're coming back. They have a trio acquisition worth 3.8 billion, and then getting into the vaccine and expanding into that pipeline as well. So as we see, the pattern is very clear. Lilly is using their capital to build optionality around high-value scientific platforms and continue to build that platform existing strategy as well. So let's move on to the next page. Okay, so if we move from facts to the strategic readout, what does this mean? The first point we want to stress here is that scale and structure. Lilly is deploying a very large amount of capital, but the capital is layered. Some deals involve acquisition with large upfront payment, but there are others that are more licensing milestones. So basically, it requires lower upfront payment. So Lilly is pursuing different bets without taking equal risks at every asset. And then the second point is that this is a thesis acceleration, not reinvention. It is actually accelerating existing pipeline using the GLP franchise cash to execute the thesis faster and then in a greater scale. And then the third point is more operating logic. So they are acquiring at maturity licensing for optionality. And then we will talk a little bit more about this different way. But then they are also institutionalized sourcing through mechanisms such as Catalyze 360. This matters because it allows Eli Lilly to be very close to the biotech community and be able to have access to those innovation and then building that repeatable external innovation engine. And then we also want to highlight that for Chinese biopharma companies, the implication is very practical. That best opportunity may not always be selling the entire company. In many cases, the more attractive route may be to become a validation partner, a platform collaborator, or an ex-China licensing supplier. The innovate structure, so we would talk about the Xingda Shengwu, that example provide a very good emerging playbook of how to partner with the MNCs. Okay, let's move on to the next page. Let's look at what happens. So what is Lilly's capability transformation before and after these deals? Before 2025, Lilly was already strong in traditional modalities, including small molecule and injectable biologics. By mid-2026, however, after all the activities, the M&A activities, company is clearly expanding into frontier modalities, in vivo cell engineering, circular RNA, DNA editing, and AI-native discovery platform. The development speed point is also very important. Lilly appears to be moving toward a model where AI, external partnership, and geographic distributed validation can compress development timeline. So we all know here, like all the company, time is money. So they all trying to shorten the timeline, the drug discovery, the development. In other words, the strategy is not only about having more pipeline shots on goal. It's more about getting the entire innovation cycle faster and more capital efficient. Then I also want to introduce this idea of bipolar innovation engine. On one side, Lilly is leveraging North America and basically the Western sphere computational strategy and high-end AI infrastructure. On the other hand, it is using the China, Asia Pacific clinical validation, patient access, and fast proof of concept generation. And then this is where Chinese biopharma companies can play a more strategic role than in the global ecosystem. Okay, let's move on to the next one. Lastly, I want to talk more about the deal structure. I like to describe this more as the difference between real capital versus wish list value. In many announcements, as we see in slide number 2, there are a lot of saying we see very large total values, but only a portion of that is paid upfront. The rest is tied to clinical, regulatory, or commercial milestones. Lilly is using the structure very intentionally. When Lilly has high conviction in a mature or a scarce asset, it is willing to pay significant upfront cash and use more of that M&A structure. Example, it includes the Centessa, Colonia. Those are more valid, have proof point already. In these cases, the upfront payment itself is the signal. Lilly is not hedging because it believes in the asset and platform is strategically important enough to own. For earlier stage platforms, Lilly often use licensing structure with lower upfront payment and larger milestone potential. This is not necessarily low conviction. It is a way to buy optionality. Lilly pays modestly to access the science today and reserve large payment for when the asset proves itself. From a capital allocation perspective, this is similar to a venture portfolio attached to a pharma balance sheet. And then let's talk about what does it mean for Chinese bio companies. For Chinese companies seeking global partnership, this has an important implication. It is critical to understand how partners interpret risk. A large total value may look attractive, but then real negotiation often should be centered around upfront commitment, proof of concept milestones, regional rights, and control over future development. Understanding the capital logic behind the structure can help companies negotiate better and position their assets more effectively. I know this is a large last page of my slides. Also want to talk about a few key takeaways. First, Lilly's GLP-1 success is not merely a commercial story. It follows with this capital allocation story. The cash flow from GLP-1 gives Lilly the ability to move faster, take more shots, and build external innovation capacity at scale. Secondly, Lilly's deal activity suggests that global pharma is increasingly interested in validated platform, AI-enabled discovery, advanced modalities, and efficiency proof of concept models. This creates opportunities for Chinese biopharma companies that can demonstrate high-quality science, strong execution, and credible clinical validation. Third, for all the China origin assets, the future opportunity may be less about being acquired outright and may more becoming a strategic global partner, especially through the ex-China rights, co-development models, and differentiated platform access. The Innovent example shows that pathway is becoming more visible and more strategically relevant. With that, I hope this study can provide a useful lens for thinking about how to think about capital innovation, global partnership model are evolving. Thank you, and I welcome any questions here.
Thank you, Stephanie. This is brilliant. That's great insight. There is actually a question online from our live stream BioValley platform. So JA asked a question. A parallel story is Moderna. Jay is very curious about your view about their capital deployment strategy after the COVID windfall.
I may have to take back that question because I need to take a look at their current strategy. Yeah, let me take back that one and then we can follow up.
Okay, thank you, Stephanie.
Mhm.
And also, actually, I have a question about, how do you see these giants, no matter Eli Lilly or other Novartis, leveraging the external sourcing and partnerships to maintain their moat as the GLP space becomes increasingly crowded?
Right. I do see that Eli Lilly is making a lot of moves to maintain the front like leadership in the GLP. But then we also see there a lot of MA, like a lot of startups also in the space and other companies, MNSAs trying to get into the space. I would say like a few things. One is that for Eli Lilly and Novo Nordisk, like we see that Novo Nordisk, they are actually revamping or scaling up their manufacturing capabilities. For Eli Lilly, we see that they're partnering with more advanced platform to develop next generation GLPs as well. So they are leveraging their free cash to gain a step before everyone else. I would say that the other thing is the commercial capability. So now they already have the brand equity. They also have the relationship with the patients and then with the providers. That's also a key advantage for those companies to maintain the front runner in the space. But I do think it is very interesting that they are not purely relying in the GLP area. So as we see that the top MAA space is oncology, which is aligned with current, when we look at MA activities in the first half of 2026, the overall trend, the key focus area is still oncology, not GLP. So as the space become more crowded, I think the companies will closely choose the bet. It is a hot topic, but does it still will give the ROI that they predict? Do we see still like I do believe the market is massive and then it could include more players in the space, but at the same time we see the companies also placing bets in other therapeutic areas.
Thanks for the comment. Your topic is very popular. You have many questions.
I know she
Box.
has so many questions.
Yeah, you have so many questions.
Ina, yeah. Look at it. The questions so in the chat Q&As, and Wenjia has several comments want to make. You may allow him or her to comment on Lily's the presentation. I mean Stephanie's presentation on Lily. Sorry.
Yeah. Considering the time, Stephanie, if you could follow up the questions in our Q&A box, that would be great.
Okay.
Yes, we have a Lija, we have a Lantin asking the questions. For example, Lija is curious about the comment on Lily's competitiveness and Novartis in GLP market, etc. So
yeah,
that
I'll follow up in the chat.
Yeah, those are very good questions.
I mean, again, thank you everyone for all the questions. I'll follow up. Thank you.
All right.
Thank you, Stephanie, for being our speaker to share the valuable insights. Yeah, I think now let's move on to take a brief moment to thank our sponsor, Bell Intron. I'd like to invite you, Dr. Gang Liu, Director of Discovery Services at Bell Intron, to present rush data, high throughput data generation for modern antibody discovery. So Dr. Liu has devoted over 20 years to biological sciences, antibody therapeutics. Now the floor is yours, please.
Thank you, host. And first, I need to thank CBA for everything that you have done to make this seminar such a success. So I just heard about some great talks which about the GLP-1 is transnational study, is FDA meetings. This kind of things are very interesting, very exciting, because that means this drug product is likely to be appear on the market very soon. Actually, that's what has happened. We have seen plenty of GLP-1 drugs, medicines that has appeared in the market. But I would say the drug dev may not reach such a long, cannot reach, cannot go such far without a good molecule for you to begin with. So what I'm talking about is a service that BioEntry has just released. We call it Rush Data. Actually, it's a high throughput data generation service for modern antibody discovery. Next slide, please. So let's talk back to the GLP-1. So why it's in gray? So GLP-1 has shown that this great success, and also actually this has changed how the whole industry thinks. So now I would say many biotechs, biopharmas, they are trying their best to searching for the next breakthrough in biology, in targets, in formats, in molecules. This also covered the therapeutic antibody realm. So as we all know, with the era of AI and also with many modern antibody discovery solutions. Next slide, please. So it's not a challenge anymore for us uh, to get a sufficient antibody molecules for us to compare to look at which we used to struggle with. So nowadays the challenge has become how can we learn and select those molecule fast, because it kind of easy for us to get thousands or at least hundreds of markers for us to learn to compare. So I would say what was once a scarcity problem has now become an abundance problem. So like which market should we move forward and which one we should not? Next, please. So to answer all of this question, actually we need uh, data. So as Dr. Her just shared, we should not hate data. Data should always be our friends. So it's data with reliable data that can help us to make decision to choose the most smart molecule for us to move forward. So Rush Data is such a package that's just released by Intrin. It's designed to transform hundreds of thousands of candidates into actionable data that can drive confident decisions. Next, please. So, but before I dive into Rush Data, so you may wonder what is Rush Data or why is by Intune. Actually, since it's founded since 2012, BioEntry has devoted itself into buildings of multiple platforms like high throughput antibody production, automation infrastructure, advanced antibody characterization, data generation at scale, and a digital foundation for modern discovery. All of this we call it in biology a smart factory. It formed the foundation for rush data. Next, please. So to put it simple, the rush data, yeah, next please. Rush data work like this way. So it's a brief workflow. Rush data begin with antibody sequences. The sequence may be from like AI tools, from phage library or single B cell screening technology, and with hundreds or thousands of sequences. You can see here, as shown here, in just 7 days from sequences, we can get as many as 3,000 per batch and antibody samples as soups that are produced in CHK1 cells. We do not use cell-free because we don't want to have any PTM gap that may lead to false positives. So in just 7 days, we can get thousands of antibody soups, and from those soups, you can get many preliminary information like its binding, its production, so that for you to achieve your rapid screening to quickly find out those that you can choose to move forward. And if you want to learn more, like how strong the binding is, their purity, their physical chemical trace, or their drug-like trace, we can in 10 days we can get purified samples. Then we can look at dose curve binding, their stability, their developability trace, and more for you to compare and choose from. Next, please. And I would say Rush data is for me, at least for me, is a perfect match for AI generated sequences because, as everyone knows so well, it's kind of easy for us to get thousands of antibody sequence by AI tools. But you may eager to know, which one are the best, at least those that you should move forward and which one should not. So with our Rush data, so once we get your AI generated sequences with as short 7 or 10 days, and also with the chew cells, there's no cell free. We can get those either soaps or purified samples in just days, and then we got action data package. So this data package can in turn feed back your AI models. Once this AI model is refined, it can once again generate, I would say, a second round of the hits. The second round can back to our rush data. So run by run again. So both your antibody sequence and your AI model may be improved and refined. So that's why I call it is rush data is kind of wet for the dry. So and once we got those data, we can deliver data your way, which means we can deliver data in a format of raw data, assay results, QC documentation, and also we can the data can be structured and standardized, and also is easy to be integrated into model training workflows, or is the data can also be friendly for API in integrations for discovery platforms. And next slide, please. And data can also be delivered, shared to you at multiple levels. No matter you are a scientist, you're a project leader, or you are an AI team member, every team can access the same package of data, same level of information to get the information that is most relevant to him or her. Next, please. So I would say data will not be data if it cannot be trusted. So with such a fast speed, such a big scale, so in every round, we include built-in controls and continuously performs monitors to ensure that data quality, data consistency, and data reproducibility. Next, please. So I would end my talk by this three-point summary. So generating antibody is no longer the primary challenge anymore. Selecting the right antibody is. AI does not reduce the need for experimental data. Instead, it raises the demand for it. Rush data, so it's not just about speed, as you can see in just 7 or 10 days, or scale, so we can handle thousands of samples in one single batch. It's more about generating the right data for the next decision. So back to GLP-1. So just like GLP-1, so every successful antibody medicines begins with many possibilities, many, now thousands or tens of thousands. So the challenge now becomes learning which one has the greatest potential as early and as fast as possible. And that's what Rush Data is designed for. Thank you so much. I would happy to take any questions.
Yeah, I think it's that we have a few minutes left. Thank you so much, Dr. Liu. And you have thank you for sponsor CBA. First
Our
of
pleasure.
all, yeah, that's very important. That we are non-profit organization. We need a sponsorship.
Yeah, that's our
To
responsibility.
formally close out our Q2 the review, this initiative start from the Q1. This second one is my privilege to introduce our Dr. Qing Li. Dr. Qing Li is the president-elect of the CBA and the head of bioengineering at Primo UX Biomed.
Primolux Biomed, yeah.
Sorry about that
Yeah.
too. Go ahead. It's your.
Thanks, Yongqiang. Yeah, thanks, Yongqiang. Good evening and good morning, everyone. I'm Qing Li, the president-elect of CBA-USA. What an incredible, successful session that we just had. First, on behalf of the entire CBA exec team, I would like to extend our sincere thank you to all the distinguished speakers from our CBA study groups, Dr. Xinrong Chen from the non-clinical group, Dr. Jian He from our clinical translational medicine group, and Mr. Rachel Jiang from our clinical record affair group, and also Ms. Stephanie Lee from our CBA Frontier Insight CFI group. Thank you all for sharing your invaluable insights and for making this webinar such a great success. And of course, we want to give a special thanks to our sponsor, BioIntron, for your continuous support over the years. So these four study groups really represent the very best of what CBA can bring us, a platform for knowledge sharing and also for the professional growth. As we look forward for the coming year, we actually have a lot of more events in plan. We are launching many new initiatives such as in-person happy hours, entrepreneurship club, and also the CFI live streaming events and many more. And the secret ingredient for making all this happen is not only our wonderful speakers or the leadership, but actually it's our volunteer team. Yeah. So every program, every webinar, every networking event is powered by the passion and dedication of all our volunteers. And that's why I'm here to call for more volunteers joining us. And as shown in this slide, CBA is looking for passionate volunteers to join our core functional teams. If you are looking to develop your the leadership skills, you want to expand your network and also work with passionate peers, this is really a chance. Whether you're a scientist or a designer or a tech person, there's always place for you. So please take a picture of this contact information shown on the screen right now and submit your resume to the contact email listed for your preferred functional team. Or you can also contact us through our official WeChat account and also our random account shown at the bottom of the slide. So we need your energy, your creativity, and also your time. Thank you again, everyone, for joining us today. Have a wonderful weekend, and I hope to see you all again at our next event. Okay, back to you, Dina.
Thank you so much, Dr. Li. Thank you for every speakers, and thank you for our incredible CBA volunteer teams, including, as Qing mentioned, CFI, social media promotion, BDIT, and logistic teams, and who made this happen. So if you aren't a member yet, we highly encourage you to join our CBA family. And thank you so much again for today, and have a wonderful night. We will see you in Q3 quarterly review.
I think that the last one is everybody encourage everybody that open the camera and the video, and then we take the screenshot. As
Great idea. Let's have all our
if you
speakers
could.
have their
Yeah.
cameras on, and also the audience. If you have any questions, if you want to unmute yourself, also please open your mic. So it's the time. Yeah, let's take our final group picture. Do we have everyone? Dina, have you taken the group picture? I saw Yongchang, you're the largest one here. So
Yeah,
I was
I did
thinking
one.
like
I did one.
everyone. Great.
Yeah, I did one. All right.
Great.
I share it with the group. Thank you.
Good night, everyone.
Thank you so much.
Have a great weekend.
Yeah, Stephanie,
care.
I will
Bye.
I will talk to you more about that. I will send you the WeChat questions.
Thank you,
talk.
everyone.
Okay.
Bye.
Sounds
Thank
good.
you.
Sounds good.
Thank
Let's
you,
connect
everyone. Bye-bye.
offline.
Yeah.
Take care.
Thank
Bye.
you.
Also,
Bye.
Richard
everyone.
and then the Dr. Gang Liu, you are awesome. It's a very good topic. Thank you. Thank you for support.
Thanks,
you.
everyone. Bye bye.
Good
Bye-bye.
night. Bye.